Module 3, working out the details of what our genes mean. This table shows the DNA content of human chromosomes. The human genome consists of 25 different DNA molecules partitioned between what are effectively two genomes, one in the nucleus and one in the mitochondria. In the nucleus, there are either 23 or 24 different linear DNA molecules, each one of the different types of chromosomes, 23 in female cells or 24 in male cells. These DNA molecules are immensely long, ranging from the size of 48 megabases to 249 megabases, as is shown in the table here on this slide. For decades, the only available map of the nuclear genome was a long, low-resolution physical map that was based on chromosome banding. Chromosomes can be stained with certain dyes, such as Giemsa, to reveal an alternating pattern of dark and light bands for each chromosome, as represented by this slide. This is referred to as an ideogram, showing a 550-band Giemsa banding pattern and constitutive heterochromatin within human metaphase chromosomes. The dark bands represent DNA regions where there is a low density of GC base pairs and a generally low density of exons and genes. Pale bands represent DNA regions where there is a high density of GC base pairs and a generally high density of exons and genes. Centromeric heterochromatin is illustrated by red blocks, and other constitutive heterochromatin is shown as blue blocks. Note the large amounts of non-centromeric heterochromatin on the Y chromosome. The short arm of the acentromeric chromosomes 13, 14, 15, 21, and 22, and on chromosomes 1, 9, and 16. Numbers to the left are the numbers of individual chromosome bands. Identifying genes and other functionally important DNA elements through evolutionary conservation. Comparison of human DNA sequences and inferred protein sequences with all other sequences in the database allow us to identify the extent to which DNA sequences have been highly or poorly conserved during evolution. Starting from a single query sequence, computer programs such as BLAST and BLAT readily allow comparison across sequence and genome databases. Over the past decade or so, the genomes of large numbers of species have been sequenced, and powerful programs allow the alignment of whole genome sequences from different organisms. Shown in this table are examples of programs and databases used to find homologous DNA or proteins from a sequence query. If a human DNA sequence has an important function, it will often have been highly conserved during evolution. It will have recognizably similar counterpart, referred to as a homologue, in other species. For genes, the homologue in other species is often an example of an orthologue, a direct equivalent that shares a common evolutionary origin with the human gene. That is, they both originated from the same ancestral gene that was present in some common evolutionary ancestor. Various resource centers provide genomic information. 3 of the more popular ones are listed below. They include NCBI, Human Genome Resources, the Ensemble website, and the UCSC Genome Bioinformatics. This particular slide shows how the Ensemble browser can be used to track gene data. The query or search term for this particular example was the F9 factor 9 blood clotting gene. The top display shows a 1 megabase chromosome environment on the X chromosome that encompasses the F9 gene, which is shown in the middle within the tall red box. The lower display shows an expanded view of this region that includes exon and intron organization and identifies two very similar protein coding transcripts, F9-001 and F9201, and one non-coding transcript, F9-002, from the F9 gene. Frequent interest is the identification of homologs across species. This slide shows the pairwise alignment of the human p53 protein and the corresponding mouse p53 protein. The protein sequence alignment was generated using the BLAST-P program as a selectable function within the homologene resource. The output lists the human p53 protein sequence as the query sequence and the mouse p53 sequence as the subject. The alignment is slightly skewed because extra amino acids can be found in one sequence that are lacking in the other. For example, the mouse p53 protein has an extra 3 amino acids at its N-terminus. The sequence from amino acid number 4 onward matches the start of the human sequence, but lacks 6 amino acids in the region encompassing amino acids 33 to 58, shown as dashes in the mouse sequence. The middle lines between the query and subject lines show identical regions at 304 out of the 393 matched positions. The plus symbol in the middle lines denotes functionally similar amino acids. They occur here at 22 positions, which when added to the 304 gives a total of 326 positive matches out of the 393. The term protein similarity is sometimes applied to this type of ratio. It depicts the human genome organization and extent of evolutionary conservation and repetitive sequences. Only just over 1.2% of our genome is coding DNA that specifies protein sequences, and another roughly 3 to 4% or so of our genome is made up of non-coding DNA sequences that have been highly or moderately conserved during evolution. Some of this conservation sequence is present in multiple copies and includes different types of repeated genes or gene families. The 6.7% of our genome that is located in constitutive heterochromatin is very largely made up of poorly conserved repetitive DNA sequences that include sequences responsible for centromere function. Transposon repeats include highly repetitive interspersed repeats such as ALU and LINE-1 repeats. It is thought that during evolution, some of these repeats contributed to the formation of new exons and regulatory elements, including some of the long, non-coding RNAs. Distribution in the human genome. More than 90% of the mitochondrial DNA sequence directly specifies a protein or functional non-coding RNA. And there is one intronless gene every 450 base pairs on average. The nuclear genome is very different. The gene density is much lower. Genes are frequently interrupted by introns, and a sizable fraction of the genome is made up of repetitive DNA, notably highly repetitive non-coding DNA. Close to 7% of the nuclear genome is located in constitutive heterochromatin that remains highly condensed throughout the cell cycle. Constitutive heterochromatin is essentially devoid of genes, but does contain a tiny number of genes that are inactive in somatic cells but can be expressed in germ cells. The remaining 93% of our genome is accommodated in euchromatin, which is less condensed and contains the vast majority of our genes. Protein coding genes are comparatively easy to identify. The most recent GenCode data estimates that there are close to 20,400 human protein coding genes. The number can never be exact because of the variation between individuals in the copy number of some repeated genes. Identifying RNA genes is difficult. 3 characteristics make this so. There's no associated open reading frame. RNA gene sequences are often not well conserved, and they can also be tiny and overlooked. That makes it sometimes quite difficult to establish the functional significance of many DNA sequences that are used to make up transcripts. The most recent GenCode data might suggest that there are close to 22,000 RNA genes, but the functional status of many putative RNA genes remains unproven. This table shows the relative distribution estimates of various types of genes within the human genome. Examples of tandemly repetitive coding DNA and clustered gene families. In A is shown normal alleles of the HTT Huntington gene have an array of tandemly repeated CAG codons in exon 1 that varies in number up to 35 repeats. Having 36 repeats results in Huntington's disease. In B, the LPA gene encodes lipoprotein A, a protein with multiple Kringle domains that are each 114 amino acids long and extremely similar in sequence. Each Kringle repeat is encoded by a tandemly repeated pair of exons, here labeled A and B, that can be present in different copy numbers. The example shown here has 9 pairs of A and B exons, starting with exons 2 and 3, E2 and E3, and continuing through to exons 18 and 19, E18 and E19. In C, the beta-globin gene family has 6 highly related genes. 4 genes make alternate globins used in hemoglobin, but the status of HBD is uncertain. Delta globin is never incorporated into a hemoglobin protein, and HBBP1 is a pseudogene. In D, the HLA human leukocyte antigen region of normal individuals has a tandemly duplicated unit containing 4 gene sequences, encoding 9 serine threonine kinase 19, complement C4, cytochrome P450, and tenascin. Subsequently, 3 of the genes became pseudogenes, having acquired inactivating mutations or having also lost significant amounts of sequence. This slide depicts examples of multi-gene families in the human genome. A processed pseudogene is a copy of a gene's transcript and so has counterparts of exon sequences only, unlike an unprocessed pseudogene, which is a copy of a genomic sequence and so also has sequences corresponding to introns and upstream promoters. Here we see several different members of gene families: the beta-globin gene family, the class 1 human leukocyte antigen or HLA, the neurofibromatosis type 1, ferritin heavy chain, and U6 snRNA. Tandem gene duplication. Gene duplication can occur after sister chromatids or non-sister chromatids of homologous chromosomes mispair so that they are slightly out of alignment. In this example, the result is that gene A on one chromatid is out of register with gene A on the opposite chromatid. Subsequent breakage of both chromatids at the position marked by the cross X and swapping of fragments between chromatids can result in a chromatid that has 2 copies of gene A. The left fragment on the top chromatid joins to the right fragment of the bottom chromatid. The exchange may be facilitated by base pairing between highly related, non-coding, repetitive DNA sequences, shown in orange boxes. Retrogenes and retropseudogenes originate by reverse transcription from RNA transcripts. In this example, a protein-coding gene with 3 exons is transcribed from an upstream promoter, and introns are excised from the transcript to yield mRNA. The mRNA can then be converted naturally into an antisense single-stranded cDNA by using cellular reverse transcriptase function. If this occurs in the germline and the transcript integrates randomly into the genome, it will most probably be incapable of expression. It will lack a promoter and will degenerate into a retropseudogene after acquiring deleterious mutations, red asterisk, bottom left. If, however, the transcript inserts next to an endogenous promoter, it may be expressed. Very occasionally and according to need, the expressed gene may be useful and be preserved as a functional retrogene, as shown on the bottom. Retrotransposons can mediate exon shuffling. Exon shuffling can be performed with retrotransposons such as actively transposing members of the LINE or line 1 sequence family, as shown here. Full-length L1 repeats have promoters and can be transcribed, but they have weak poly-A signals, and so transcription often continues past such a signal until another downstream poly-A signal is reached. The resulting RNA copy contains a transcript not just of the L1 sequence, but also of a downstream exon. The L1 reverse transcriptase machinery can act on the extended poly-A sequence to produce a hybrid cDNA copy that contains both the L1 and E3 sequences. Subsequent transposition into a new chromosome location may lead to the insertion of exon 3 into a different gene. And this concludes Module 3.